Hi, everyone. We're just going to give it a minute to let everyone into the meeting room, and then we'll go ahead and get started. All right, I think there's quite a few people in the room now, so I think we can go ahead and begin. Hello, everyone. Thanks so much for joining our microtomy workshop today. The focus of today's webinar will primarily be on, quality tissue preparation to ensure artifact-free staining. And I'm really excited to have Laura Taylor from National Health Service here to lead the workshop. But before I go ahead and pass it off to Laura, as always, I just want to do a short company introduction to familiarize everyone with our company, Protein Tech. So, Protein Tech is a leading manufacturer of antibodies and immunoassays. The company was founded in 2001, and at that time, it was primarily a supplier of antibodies and immunoassays. Because we've been in the game for over 2 decades, we have a really large portfolio of antibodies that cover about 2/3 of the human proteome. But we also offer assays such as ELISA kits and IHC kits, as well as cell separation kits and antibody labeling kits. The company then later expanded to include more product lines. So we now also have our humankind branded cytokines and growth factors. And all our proteins are going to be expressed in human HEC293 cells so that you can be confident that they have the native folding, post-translational modifications, and glycosylation that you would expect to see in human proteins. And that ultimately enhances their performance and increases their bioactivity. We also have our Chromatec branded nanobody based reagents. So, nanobodies are about 10 times smaller than traditional antibodies, and they can bind to their targets with really high affinity, which makes them ideal for several applications such as immunoprecipitation, immunofluorescence, especially if you're doing things like super resolution microscopy, as well as protein purification. And then lastly, we also have a subsidiary of Protein Tech known as Protein Tech Genomics. And Protein Tech Genomics manufactures antibodies that are conjugated to oligonucleotides for single cell RNA sequencing. So, as a company that is driven by scientists, we're here to support our researchers and scientific community any way that we can. So, our award winning products are self manufactured, and we offer the largest in house portfolio of antibodies for the human proteome. For convenience and testing, most of our antibodies are also offered in 20 microliter trial sizes. We also know the importance of product validation, and we offer open access validation data. Whenever we are testing new products, we do validate with relevant sample types, appropriate experimental conditions, and using knockout knockdown models whenever possible. Also, as antibody experts, we do have in depth knowledge of immunoassay applications, and we can offer outstanding technical support for our customers. We also stay on top with cutting edge technology, such as our proprietary antigen specific B cell cloning and engineering platform, which we use to manufacture our recombinant antibodies. And then lastly, because we are a global company, we do have 7 inventory sites and manufacturing and research sites on 3 continents. So we are able to offer next day delivery to many areas in the world. As a company, we also measure our success by our customers' scientific success, specifically by helping them publish impactful research faster. So, this graph here on the left shows that over our company history, our products are featuring in more and more publications each year. And as of now, we have over 300,000 publication citations, making us one of the top 5 cited antibody companies and also one of the fastest growing companies in product citations. I also just really quickly want to highlight that as a company, we do also strive to make the searchability of our products on our website as user friendly and informative as possible. So this year, we have launched some new updates on our website to help our customers select the right antibody. This includes our unique 3D epitope viewer that allows you to interact with epitope structures and explore antibody binding sites so that you'll know exactly where our antibodies bind to your protein of interest. We've also introduced advanced filter options to help you find exactly what you need, such as our immunogen region selector toggle, which allows you to filter for antibodies generated from your region of interest. And now you no longer have to filter through all the variations of an antibody from the search results. You can view all formats of an antibody, including conjugations and buffer formulations, on just one page. And the goal for this was to ultimately streamline your process for antibody selection. So with that, I think we can go ahead and kick off the workshop. So I would like to introduce our speaker for today, Laura Taylor. So Laura is a registered biomedical scientist and is currently working on her specialist portfolio in cellular pathology. With 10 years of various lab experience, she is familiar with a range of laboratory techniques. She also completed her master's in molecular biology and biochemistry and is a keen science communicator. So with that, I'd like to welcome Laura, and I will go ahead and pass it off to you. Thanks so much for being here.
Thank you very much. Let me just get my screen ready. Right, here we go. Is that, can people see that? Or is it all good?
We're seeing your presenter view now.
Presenter view, excellent.
Yeah.
So that's all okay?
No, we're seeing like
Oh,
your with the notes.
oh no.
Yeah, I don't know why I did that. Didn't do that during the practice session.
Hmm.
Um,
Bear with me one moment then. Let me try and do. Apologies, everyone. Right. Oh,
okay, that looks good.
okay, can you see this but without any notes?
Yes, no notes.
Excellent, brilliant. So, hi everyone, my name's Laura, my pronouns are she, her, and I am a biomedical scientist working in the NHS in the UK. So today I'm going to talk to you about immunohistochemistry. So what steps there are in it, including what issues can arise at every step and something you can do to fix those issues. How to prepare sections for immunohistochemistry, including talking about blade selection and the best way to set up your area. And I'm going to do something that I like to call cause and a fix, where I show you all the different issues that can happen with immunohistochemistry and the fixes that you can do to rectify those. So this presentation is aimed at those of you who might have not done immunos before and would like to sort of understand what you're getting into, or people who are going, my goodness, I've been doing this for a while and I'm getting a lot of issues. What are some of these common issues and how can I fix them? And I'd also just like to say before I start, so this is based on my own experiences with the equipment that I've used in the lab. A lot of NHS labs are lab management service contracts. So if you're in somewhere that has something like that, or you're in a specific lab, before you go in and go, I want to change all of our protocols because I've been to this presentation, just double check with your line manager first and like the sort of things that you actually do in your job at work. So very basic, but I'm just going to start off with what is microtomy, because this is what we're going to be talking about. So immunos, typically you tend to use either microtomy or you can use cryotomy if you're using fresh tissue. So microtomy is just the cutting of tissues using a microtome. So this is a microtome. You might have used one of these before, you might not be aware of what they are, but it's done to produce slides for staining, and then you examine them and diagnose from these slides. So this is just a little example of what a microtome can look like, with just some annotations of what it is. So you've got things like your chuck and your clamp in the middle. So this is where the blocks go. You've got your blades with your blade guard, and you've got things like your thickness adjustment of what you want those blocks to look like and your sections to come off as. You might use an automatic version. These are the ones that I've been using for the past 5 years. So this is what I'm most common with, but you can also get a cryotomy, which is used essentially just a microtome in a big freezer, is what I like to call it to people. And that's something that you use to cut fresh sections. So with microtomy, you're typically using embedded blocks that are in wax and they have been fixed and processed. And what is immunohistochemistry? So the other part of what is going in hand with this presentation. So this is a staining technique, and it involves identifying your target antigens in your sample, and it's using antibodies to bind to them. And this can then be visualized. So it can be visualized typically with this brown pigment that we can see on this image here. Sometimes you can use fluorescence, or sometimes people use a red pigment. They might use a combination of both. And it's used to identify the disease, specifically where the disease is coming from if you have a cancer of unknown primary. It's really useful for staging and grading of the cancer, and deciding what treatment or options there are for the patient as well. But you can also use this in research. I'm just coming at it from a patient view just because that is what I'm in healthcare. So you might be thinking, let's go straight into immunos, but I'm not going to go into that straight away. We can have a few slides on actually where to go from your fundamentals. So you're not going to get good immunos if your blocks aren't good to begin with. So for example, you need to ensure that the samples requiring defect calcification are soft enough to cut on a microtome. You can use different blades to cut hard tissues like that, but just please be aware of the effect of decalcification solutions on immunos. Sometimes they can really degrade the areas that you're looking for. So if you have got something that you think this is really difficult to cut, but it needs a bit of decalcification, but it's likely going to go for immunos, maybe just have a little bit of chat with your supervisor or who you're working with to see how they'd like you to go and proceed with that. Block thickness, if your samples are so thick and fatty, for example, the breast tissue that's been cut like a door wedge, that might be really difficult to cut and be reprocessed. And especially in the area that I'm working in healthcare, that can cause really horrible time delays, and we need to get those results out for the patient as quickly as possible. And also just embedding samples, sorry, in a line to avoid scores from calcified areas. So this is the image that I'm showing you on the right hand side. So if you imagine that these are 3 pieces of tissue all in a row, and you can see your top one has got a little bit of calcium on the top. Now, if you're cutting that on a microtome, when you're cutting it, it's going to go all the way through the rest of your tissue and leave a horrible score throughout them, and might actually obscure the lesion, the rip that you get, something like that. And so we don't want to embed things like that. We'd actually like to embed them slightly on a diagonal. And this just helps be able to get like that nice section. And if you have got some calcium in one of those bits, it's only going to affect this one area. It's not going to affect all of the different areas of the tissue. And mounting, so picking up the tissue, just some things to consider. So for example, your slide type. So typically for immunohistochemistry, we use charge slides. So this is because it adheres the tissue to the slide easier. This is because, especially when you're using an immuno machine, it's a lot of a harsher process. So sometimes those tissues can lift and they can come off. So what we want to do is make sure we're using a charge slide to keep that tissue on the slide. It's also useful in normal histology for things like keratotic tissue, sometimes samples that are a bit bloody. Those can also be really beneficial from picking up on a charge slide. Your orientation as well, I always say to people, pick up the tissue, not the wax. And this is something you can see on the right-hand side. So people will go, well, you've cut me a lovely little rectangle there. I'm going to pick up the rectangle. But actually, I'm not bothered about these bits of tissue at the side because they've not got any tissue in them, these bits of wax, sorry. So what I want you to do instead is concentrate on picking the tissue up all in a row. And then you can just have these bits of wax hanging over because we're not worried about them. Whereas here you can see that there's some bits cut off on the side. And if those are your areas of interest in immunos, that means that the person diagnosing or you yourself as a researcher looking at them isn't actually going to be able to see that the areas in that tissue. And also, we're now moving towards digital pathology. So we scan all of our slides. So just making sure that you're leaving a bit of an area around the tissue, otherwise your scanners won't pick that up. And just keep your biopsies all in a row. Up here for a start, the top one, I know on our scanners will get cut off, and they might just get a bit confused about what's level 1, 2, and 3. So this is an example of biopsies. Whereas you can see here, these are all straight in a line after each other, and that's a better way to pick up the samples when looking at biopsies. And here are just some like real life examples of this. So you can see here, it's just the tissue's been completely cut off in a couple of different areas. So this is the digital, part B is the digital image of part C, the slide, which is the physical glass slide. And you can just see, it's just, the section is just a bit big. It definitely could have been moved over a bit more to the left and you would have got more of this tissue on. This could be due to the tissue blocks that you've taken in the first place, but also this definitely could have been picked up better. And in part A, again, you've got the epithelium missing. That is typically the most important area that you're looking at times, especially with immunos. So you want to make sure that you've got that on your slide, and you're not able to see it here. And then part D and E is just an example of the picking up the tissue, not the wax that I mentioned earlier. So part D, they've picked up the wax, and you can see as much as it has fit in, if it was a little bit bigger, it might be getting cut off, and also how temperamental your scanners are might also cut it off. Whereas part E, what they've done is they've got everything in a nice row straight down the slide. So labeling slides, again, this is based off something that we do in the lab. We scan everything. So every single slide that we have in the lab, it's got like an individual barcode on it. It's called a Cerebro tracking system. And it's just something we do to track the blocks and slides around the lab. But the tracking system isn't compatible with our slides, with our immunomachine, which needs different labels to put on. So we have to hand label every single slide on the immunomachine. The positioning of that labels is really important, just because if you put your slides into the immunomachine drawers and it's a little bit skew-if, or it just looks a little bit just doesn't look great, it's hanging off, it could affect the slide. You could get some lifting, and that could cause a bit of background and just get your immunos to not look as nice as you want them to. This example is where the label has just been put too far down, and what it's done is that the cover slip has then gone onto the label and it's lifted up a bit. Depending on how fast-paced your lab is or the workload, this could then be left and it could dry out, and what you'll get is some really not nice, just dry, crispy tissue at the top of your slide here. This is the control, so it's a little bit better, but still not ideal if it was the patient tissue. But you can see here that you can literally got the mountain coming up, and you've got your sample there, which will then dry out. So just make sure you're labeling your slides okay. So just some considerations from your blades that you are using. So this is really important if you've got lots of different kinds of sample in your lab. I've always worked with low profile blades, and it's what we use personally for immunos. So your high profile blades, they actually have more height, hence high profile, and they're the ones that are used in cryostats, and they're generally used to cut a thickness of 4 to 15. And then low profile blades, like what I use, they're best on rotary microtomes, and they cut a little bit less, like a bit smaller than that. So this is just something that to consider when you're thinking about what immunos you're cutting. You might have a cryostat specifically used for cutting immunos, depending how big your lab is. So just make sure that you've got the right blades to use, and make sure that it's positioned properly. So that annotated image of the microtome earlier, you want to make sure that everything's parallel. You want to make sure that your knife angle and what they call it clearance angle, make sure it's proper. So just this down here, making sure that you're getting those nice sections when they come down, because too close or too far away, you could get something called thick and thin, which I'll discuss later on. And you could just get some issues with regards to what your samples look like. Like I've mentioned here, chatter, and also chunking out the material, which you don't want to get rid of. So just something to consider when you're thinking about what you want to go into. So here's your setup. So this is where I do my immunohistochemistry at work. So we want the water bath to be at 45 degrees. This is just because you want the wax tissue to be able to smooth out all of the wrinkles in the tissue, but you don't want it to be so hot that your wax is melting or your tissue exploding. If you have got any particular tissues like breast tissue, that's a bit, it's a bit fattier, just put a little bit of cold water in your water bath. It'll bring that temperature down a bit, and you might find it easier to pick up the tissue. We actually soak all of our blocks, and this is something that, from my experience, not loads of labs do. So because of the harshness of the processing, so we put all of our biopsies in with the routine samples on a 12-hour cycle, purely because it's just what works best for our lab. But because of that, our biopsies can get a little bit dry. So from this, we just need to introduce a bit of that water again. So we use some water on the cold plate, which you might be able to see here in the background. Some labs use like slush, like ice trays like that. And that just helps remove the chatter, which again, I'll show you some pictures of later. Hot plate, you want it to be at 60 degrees Celsius, and that's just to make sure that you're melting the wax properly and getting it adhered to the tissue, adhered to the slide, sorry. And what you also want to do is, for immunos, we tend to keep them for 30 minutes on the hot plate. That doesn't include our cell lines, so our HER2 samples, and we've just started with our ERs as well, because we were finding they were lifting so much. We actually put them in the oven at, I think it's 60 for 60. I think we do 60 degrees Celsius for 60 minutes. But again, check with your lab that you're working at to see what works for you. But that's something that I would consider if you're finding a bit of a difficulty with regards to your sections lifting. And then I've put doing the immunos, then the levels. So this again, I'm coming at this from a healthcare perspective. If you're getting some requests for immunos from the consultant, they've gone, I think there's something in this patient sample. Make sure you're doing the immunos before you're doing the levels or whatever order they've requested it in. Because if you can imagine they've gone, oh, you've given me a skin biopsy and you've given me 3 levels in this tissue. I can now just sort of see something here. So I think I'm going to get an immuno, but then to assess how deep it is, I'm going to request 4 more levels. If you go ahead straight away and cut your 4 levels straight away, you might have cut out that area of interest that they've asked for the immunos, and that might affect the patient diagnosis and they might need to get rebiopsies. So just double check that that's what you're doing and make sure that you're not cutting anything out of importance. Also making sure that the microtome is set up correctly. Please make sure it is finger tight. We don't want people absolutely ramming their whole body weight behind it. You'll just damage things and it'll just make sure it's too tight and you might get it'll just cause issues when you cut in, to be honest. So finger tight, don't throw your whole body weight around it, and just making sure the alignment's correct. So what I was saying earlier about the clearance of the clearance angle of the blade and things like that. So now I'm going to talk you through the whole areas of sample preparation to diagnosis, essentially with immunos. And what you're going to get is a few bullet points about the information, and you're going to get a little box of the issues that could arise from when this is done incorrectly. So the 2 main ways that we do immunohistochemistry is either rapid freezing and using a cryostat, or fixing the tissue and creating these paraffin-embedded blocks. The tissue must be rapidly preserved to avoid the loss of proteins, because if you let them degrade, they might not show up when you're doing your immunos. This is, for an example, an issue that we had with regards to samples like breast samples weren't coming to us in time. For those who don't work in a healthcare histopathology lab, when you get a breast sample, you tend to bread slice it. So literally slice it as you would bread to get the fixative inside it all. Because if you can imagine that there's a tumor in the middle of that person's breast and you're just putting formalin on the outside, it's not going to get fixed and those proteins could degrade. So we need to make sure the breast is coming to you as soon as possible at any sample to make sure that it's fixed optimally, just so that the immunohistochemistry is able to perform as best it can. So the crosslinks, however, formed in fixation can block the target antigens. So optimize your protocol that you're using for this. So as much as we don't want things to be underfixed, we also don't want things to be overfixed. One specific example of this is with regards to estrogen, progesterone, and HER2. In breast cores, you want them to be fixed for over 6 hours, but ideally less than 72. And this is just because it can really cause an issue when applying your antibodies and getting your results. So some issues from here, like I said, not fixed enough and causing false positives, or fixed too much, which will block the targets that you're actually looking for because of the crosslinks that are formed. So here's just an example of some poor fixation. So several studies have shown about the delays in ER and PR, your estrogen and progesterone, can cause issues. So these top 3 on your left-hand side, A to C, that's your ER with no fixation delay. So that brown signal that I've said, that's some immuno show, lovely. It's absolutely fine in all of them. It looks great. However, on the bottom one, it had an overnight delay of being put into formalin, and you can see the signal's not that strong. So if you can imagine, and the same for the right-hand side, it's just the progesterone version of this. So if you can imagine, that could really affect a patient's outcomes, or it could affect the quality of what you're doing in research. So just make sure that things are fixed properly before you put them in. Even if it causes a bit of a delay, it'll be absolutely fine. So just make sure there's no delay with your fixation, get it fixed straight away. Obviously, this doesn't count for your things that you're doing fresh when you're using cryostat. But yeah, this is what we do for immunos when we're fixing the tissue. So your steps for immunohistochemistry next, once you've got your sample and you've cut your sections, you've got them on the slide, you need to remove the paraffin, and that is known as antigen retrieval. So you typically use heat or an enzyme to unmask the antigen needed for detection. And this is the thing that might have been blocked by fixing the tissue with those crosslinks. So I know that we use heat. It just stays on a hot pad and it melts everything out and it helps to unmask the antigen. But also you might want to block non-specific sites to avoid your background. So one thing you can do here is use a buffer. So an issue, if your blocking step is too little or not included at all, you could get that background because you've got these things bound to it anyway, which is non-specific. And if your antigen retrieval time is too short, some antigens may be blocked and you might not get the sort of the strength of the color that you need and the antigens that you want to look at. So just an example of antibody selection here. So when you're having a look at purchasing your antibodies or having a look at what you want to like target, there's lots of different options. So typically most things will need a primary antibody, but some will need a secondary antibody as well. So your primary antibody is the one that detects to the target antigen. If your label is attached to this antibody, it's direct immunohistochemistry. If you use a secondary antibody, it's indirect technique. So your secondary antibody will bind to your primary target antigen, just make it a secondary. So you could use a monoclonal antibody. This recognizes a unique antigen, so it's really specific. But what happens if your tissue's been fixed too long and you've got all these crosslinks and actually your target's hidden, or you've not used the proper you've not used some buffer wash and actually everything's blocked, then what do you do with that? So just some considerations. A polyclonal antibody could be a better choice when the protein target is expressed at lower levels. These recognize multiple different epitopes and they offer greater sensitivity. So you could have some issues. So you cross reactivity between antibodies and tissues. That's why you want to make sure they're from different species, what you're targeting. You might get some background due to no rinsing. Typically in IHC, you have lots of rinsing steps as you get rid of everything that's unbound to get it ready for the next step. It just helps with making it look a bit nicer. You could have a weaker antibody visualization due to it not binding at all. And it's been suggested that maybe going at longer times, but for lower temperatures, this could really help with that binding. So your detection and visualization. So you can counter stain this, and you typically use hematoxylin like you would in a traditional H&E stain in the lab. And then when you want to get your images, you're either going to use colorimetric detection or fluorescence. So colorimetric is where the bound primary or secondary antibody, they'll have a coloured label attached to this, and it'll produce a precipitated product when it's activated by an enzyme. And then you could use fluorescence, which is a fluorophore is bound instead of the dye, and it fluoresces in the presence of a specific wavelength of light. So it completely depends what you want to do, which version you'd like to have a look at. But there could be some issues. Again, background, I've mentioned it pretty much as an issue in every single step so far, but making sure you're getting your washing done to get that background got rid of. But also a lack of detail in the counter stain as well. Is also quite possibly an issue. So that'll be with your counter stain of choice with your hematoxylin. So control tissue. So typically every slide that gets produced will have a control on it. I know in the lab that I do, it does. So what you need to think of is what's the best control for each antibody. So this is an example of the drawer of antibodies slides that we have in our lab, control slides, sorry. So you can see that for skins, I've got some that are really good for SOX10 and melon. I've got CEA with some colonic tumor. We've got some prostate that's positive for PSA. You can see loads for appendix. So this is really good because it means that rather than cutting one for each one of these different tests, you've got one box that'll do everything. So that's what I would really recommend as well, is just having a look at what you could sort of hit with the most. Especially if you're in a place that you struggle to get control tissue with, I'd really recommend it. But what does the antibody provider recommend? So whoever you get your antibodies from will typically give you a data sheet on what antibody, what control tissue they recommend that you use, and the protocol that they recommend that you use as well. But does this actually matter? So this is very specific to the UK, but in the NHS. But for example, like our governing body is UCAS, and if we're using a control tissue, like a different type of tissue than what the provider recommends, we sort of need to justify that to our governing body for when they come around and inspect us. And that could be, for example, we don't have a lot of this, the specific antibody that the provider recommends, but we've got loads of control tissue from a different kind of tissue. And it just makes a bit more sense for us to do that. So that could be one example. How do you test if your control tissue is working? So you use a first and last section and then quality control this. And I think I have an image of that is in a couple of slides time. When you're cutting your control tissue, make sure you're picking up in the right areas and allow them to dry overnight. We also keep the cell lines in the fridge. This is just because the storage of it can have an effect on the antigenic potential. So cutting how many you need, when you need, and keeping them in the fridge can prolong this. And you can also bake slides in the oven if needed. So we typically either do an hour at 60 or we do overnight at 37 degrees Celsius. So just double check what your specific control tissue needs for your immuno runs. And then, yeah, as well, you might work in a really busy lab and not have time to be cutting loads and loads of control slides. So you might not cut loads that often. We typically tend to do like all of ours in a batch a week as and when we have time. And it lasts us like a couple of weeks, depending what it is. But you can see here, this is an example of two different control tissues on the slides. So you can see here the red box is where the control tissue is, leaving me space in the purple box to cut up my sample. Left-hand side, I've just got to hope when I pick that slide up that the tissue I'm looking at is quite small because there's not a lot of space left on that slide that's been left for me. Whereas on the right-hand side, my tissue is at the top, nice and neat, and I've got loads of space on the bottom to be able to pick up my test tissue. So just consider this as well. Make sure your blocks that you're using for control tissue, cut them down if it helps. And this will just help you a little bit with making sure you've got space for your test slides. This is an example of your the SOX 10 first and last control. So make sure your control slides are fine. So you can see here, this is the first on the left-hand side. And you can see this positivity in the epithelium. Whereas on the right-hand side, yes, we have a little bit further in the tissue, but the epithelium is cutting out. So it's just something to make you aware of. Always check your first and last and make sure that that block is still expressing these antigens and that it's all looking okay. So now we get on to the issues and what can go wrong. So you might be thinking before I've even got to my immunos, I'm having issues cutting the sections to begin with. So you might be thinking, my goodness, my sections are all wrinkly and they just look not great at all. So this could be because your block's too warm, so get it back on ice. Your knife angle might be causing issues, so just making sure everything's finger tight. And your water bath might not be warm enough to spread those tissues out. So just double check about your water bath and make sure it's at the right temperature. Just to go straight on to the issues with water baths on the bottom here, that's an example of what I call an exploding section. So you can see here the tissue is literally exploding on the water bath. So that could be because your water bath is too high. It could also be that you're putting too many sections on the water bath and it's taking you way too long to pick them up. Or your block could be under processed. So again, that was an issue I mentioned earlier with regards to like tissue thickness. So take your section quickly. You can reprocess or re-wax the block if needed, or like put a bit of cold water in your water bath to try and get that tissue up. You could also cut it a little bit thicker, but again, you don't want it to be too thick because it'll affect the quality of your sections. Here you can see it's got your tissue is compressed. So this could be because of a bad knife tilt angle. So make sure you're correcting the blade angle and soaking your block for less time so the tissue doesn't compress. You also need to make sure you're picking the optimal section every time that you're cutting your sections for immunos. So your first couple of sections are likely going to be too thick and your last few are likely going to be too warm, so will produce a wrinkled section. So somewhere in the middle, about 3 to 5 is typically okay. Just make sure you're getting your optimal section. So section issues, these are before and after some recuts. So on the left, you can see this is some tissue ripped. This is because it's a bit of muscle in a colon. So the tissue was ripped because it was soaked for way too long. So the tissue just went a bit mushy. So just soak it for a bit less longer. This is the same. I tell people to soak uterus as well for a little bit less long time as well. On the right hand side, you can see an incomplete section. This is just because you've not gone deep enough. But I would always say to double check what needs to be put back for recuts. I'm really fortunate that the lab that I work with is quite small. So if I've got a section where I'm like, I'm not 100% happy about this, but I know it will cause a delay to the patient if I hold it back, I can just go to the pathologist and say, can you diagnose from this or not? But again, just double check where you're working, or you might be working in research, so you need to make sure it's right every time. It's completely up to you and your lab. But this is, for an example, incomplete section, the epithelium is missing. So it went back, had a recut and a deeper section, and now the epithelium is there. And as this is something that can be targeted quite a lot during immunos, it's really important to make sure you get that complete. Here's some specifically from immuno. So these are some horrendous folds and some awful scratches. So folds over the epithelium, which you can see here. So this is possibly because you've over soaked it, which you might have a crease on the section when you're floating it out. Just recut it, make sure that you've got a really nice cool block for cutting. As well, you could stretch the sample out on the water bath. So if you see over as a fold there, you could use your forceps to like tap the fold out. But this could also cause the sample to get ripped in other areas. So just do what you're comfortable with, but make sure that, making sure that you're getting your sections without any folds on. Here you can see that there's some scores. So this is likely because there's either a bit of calcium or keratin or something here, which has caused a score down the tissue, or your last section, the last block that you cut has some keratin and calcium in, and you've not moved your blade along. So anytime that you cut a blade that's absolutely horrendous, move your blade along to get a fresh section when you're cutting your next tissue, because you don't want all your sections that you cut afterwards to have the marks that you've got from the other tissue that you've done. Also could just be due to the tissue type as well. And these scores as well. So again, just new blade, making sure that your water bath temperature is okay, and making sure that you've got just a nice setup for your immunos. You could always decalcify the calcium, but again, that could affect your immuno, so just take care if you do choose to do that. If your tissue is really bloody, you could cut it straight from warm water. So I soak mine in the water bath for like not a lot of time at all, put it back in my freezing cold water bath, and then normally it tends to cut okay. So one of the most common areas I would say with immunos is lifting. So here are some examples of some lifting. So you can see here, this used to be a breast core, and now it's just the outline of a breast core essentially. And here I think was some skin, and it just completely lifted, and this was also some skin that completely lifted. Again, it could be due to the tissue type, so that could be why the tissue is not adhered. So just get it hot plated for longer. We received samples from somewhere that was putting albumin in the water bath, and it was just not great. It caused a lot of issues with the slides that we were getting, including some lifting. So yeah, just double check what they're doing as well as what you're doing. Using an adhesive slide, so that charge slide we mentioned earlier, and you can also bake the slides in the oven to get them on properly. So just one example. Oh, and also just have a look at the protocol, because if you've got some really aggressive washing steps, you might have a bit of an issue. But if you need them to get rid of the background, then that's when you'd go back to your sample preparation and make sure that things are like really baked on there and really stuck. Some more section issues. So this is thick and thin section, or as we like to call them, the Venetian blinds. So this is where particularly an issue in hard or dense tissue, such as uterus or bone, or tissue with different densities like brain. So if you just make sure that you soak the block for less time, make sure it's super, super cold, and go slower with cutting these sections, but not so slow that you cause the section to then be completely thick. So again, if you're using a cryostat for your immunos, this might be something to keep in consideration. But if you cut a little bit slower, or trim at a lower size, and just soak it for a little bit less time, just making sure that your microtome is, again, finger tight. I'll keep repeating it, so it's not loose, because that could also cause those vibrations. This is an example of trimming holes or moth holes. Again, if you've trimmed too aggressively, this is something that I, rather controversially as it seems, I don't sit down when I do my microtomy. I stand up, and I have my microtome at like a 45-degree angle, just because I find it a lot easier for me personally, for my back and things, but also because when you're sitting straight on and you're sitting down, you're having to lean over quite a lot to see the distance of your block to the blade. And that is what I've noticed a lot of the trimming holes comes from the people who do that, because they think, oh yeah, that's fine. They go down to trim it and it just goes woof, it gets rid of a lot of the front of their sample. So this is just an example, and one little tip that I will say, I would really recommend that people do. So yeah, you've just trimmed into aggressively. So trimming slower. If this does happen, you are able to then trim in a little bit more and smooth off that section. So if you do accidentally trim in too much, and you've like scuffed the front of the block, just you're able to redeem yourself and just trim it in a bit more to smooth it off completely before you go to soaking it. This is just an area of, I've called it background. I'm not 100% sure what I would call it properly, but essentially the section's been picked up too close to the edge of the slide. So then when it's like gone for on the immuno machine, it's got this like dirty line in a way. So it just doesn't look great, and it's also sort of looked a bit naff during cover slipping as well. So make sure that but if your area of interest is present and it's these bits that you're more bothered about, that you're looking for your diagnosis in, and these bits, then you won't need to recut it. So again, just double check with who has requested those immunos. Chatter. So this is something that is again that I mentioned earlier. So this is because you've under-soaked your blocks because the process is really harsh and you've cut too many sections. It also could be because you're cutting too quick, sorry, but it is most likely because you've under-soaked your sections. So just putting your sections back into water for a little bit will really, really help. You'll be able to see, because as you're pulling your sections out of the microtome, you'll literally be able to see the tissue turn white. And that just means that your tissue is way too dry. But make sure you soak them for a bit longer. But if your water is freezing, then it won't soak at all. So you want like cold water, but not absolutely freezing, and it'll just help to get rid of that chatter on your sections. So some section issues of artifacts. So top left is squames. This is just your squamous cells. It's because you've not washed your hands and you've touched all over the sides. So now everything's got these little floaters on them. And it's not ideal, and it could cover the areas of interest. So avoid touching the slides when you're picking up. Just touch the area at the top that you're going to put the label on them. Any of these issues on this page can cause the lesion to be obscured. So yeah, you just wanting to avoid that as much as possible. Carryover. So this could be because there's tissue on the water bath. Carryover is like particularly bad because you might have tumor contamination. So this could be the actual patient that you're looking at or the sample that you're looking at, and that has tumor in it. But you might go, this also has tumor in it and it's the same tissue type. So how do you know what patient that's come from? If you've got carryover, you might end up giving like the wrong results out. So it's really good to make sure that you keep, I put replace water weekly. That's just because of the amount of sections we do. But you can replace the water, skim the top of your water bath with tissue to get rid of all the little bits that stick around and float, and just make sure you're keeping everything nice and clean. Here, if you can see it, is an example of a hair or a fibre in the water bath. You can recover slip it if it's obscuring the lesion. But if not, then it should be okay. But yeah, not loads that we can do to avoid that, other than again skimming the top of the water bath. And this is a fingerprint which my colleague did, which I like to show her every now and then. That I just think is quite funny, and she just just touched the slide by mistake. So it's just caused a fingerprint over the tissue. So this is again just restaining it, and it didn't cause much of a delay at all. So that wasn't too bad. So another issue is having water on the sides. So this could be because when you've picked up, you've got loads of bubbles in the bottom of your water bath, and it's sort of kept on there. You might have on your slide, you might have put your slide down on the water bath way too early, and it's caused you to have lots of like bubbles still remaining on there. It could be an issue with the washing steps. So you can see here the top two slides is the same image. These are some prostate cores, and you just saw it here. We've got this like background of this like little brown patch where it looks like water has stayed on the slide during staining. So it's not been washed off, and it's just made it just not look particularly great. This again is another version of like a bubble or water on the slide, which has caused this like area to be a bit of a bubble. This has caused issues with scanning and again with cover slipping. So redo your samples if necessary. Ensure that the slides are dry before you hot plate them, like really dry before you hot plate them, especially if you're putting them in the oven, and make sure that your wash steps are appropriate. Another issue is background, and there's lots of causes and fixes here, so I'll quickly run through them. So you might have a blocking of the non-specific binding might be insufficient, so increase your blocking incubation period and change your blocking agent. If you're using an enzyme conjugated antibody, the endogenous enzymes might be active, giving false positives. So this completely depends on the antibody, which your data sheet will have information on, and what to do to fix this. The secondary antibody might be binding non-specifically, so have a run of the control without a primary to see, so that you're able to check the secondary antibody. Your primary antibody concentration might be too high, so therefore reduce your concentration. Your tissues might not be washed enough at lots of different steps along the process, so it just increase your washing times. And this is an image of an ER control where we had, for some reason, some background issues. So this was meant to be very positive, little less positive, not very positive, and completely negative. And you can see every single one came out positive. So we just had to have a bit of an investigate to see what was causing that. So this is my last slide. So just again, just some specific issues regarding background on HER2. One thing that I will say, just coming from just sort of the background that I'm at, is that we just try and get things out as quickly as possible without delays, because you want those patients to get diagnosed. So you might, like, throughout this, you'll have heard me say, double check with the pathologist if they can diagnose from it, or if the lesion is still present, then it's okay, you can send it out, things like that. Again, just double check with who is in charge of your lab. This is, again, just something that I do to get those slides out to make sure the patient's diagnosed, rather than holding everything back for a 4-hour immuno run and then scanning overnight and things like that. So this is just one thing that I've mentioned. And this was just something that came up a couple of weeks ago. We got some background on our HER2 cell line. So you can see that there is a presence of cytoplasmic staining on these slides, but the stain should be a membranous stain. So you can see here it's these like sort of little like granular areas that we were just sort of struggling with, when actually this is sort of what we're most likely to see in the HER2 stain. And this is a bigger image of it over here. So this got sent out because the correct parts were stained. So a negative slide was also used every time we do a HER2. So we decided it's absolutely fine to send out, and it was just sent out to avoid delay. So that is the end of my presentations. I apologize if it was very, very quick. Lots of areas to cover. So hopefully that you've learned something today. And I see that there's lots of questions that I'm happy to answer. And yeah, I think that's it for me at the minute.
Awesome. Thank you so much, Laura. That was a wonderful talk, very comprehensive. As you mentioned, a lot of information to cover, but I think you did an excellent job. We've been getting a lot of positive feedback and lots of questions, which we will hopefully get to some of them. We probably will not get to all of them, but I do want to shout out. I noticed in our audience today, there is someone who's been going around answering questions. So big shout out to Robin. Thank you, because we will not be able to get to everyone, but we can always answer questions via email as well. But before we start the Q&A, I just want to share a few product slides, and then we'll go ahead and jump into the questions. All right. So before we jump into the Q&A, I just want to share a few product slides that might be interesting for those in the audience that are doing IHC. So at Protein Tech, we do have a very wide range of products that are designed to optimize and simplify your IHC workflows, including over 12,000 primary antibodies that are validated for IHC. Many of our antibodies are also offered conjugated, so you can get them conjugated to biotin, or if you're doing fluorescent IHC, we do also have antibodies that are conjugated to our in-house Coralite dyes. In addition, we also have secondary antibodies, including fluorophore conjugated, biotin conjugated, and poly-HRP secondary antibodies. And then aside from our antibodies, we also offer kits for IHC, which we'll talk a little bit more about in detail on the next slide. And we also offer accessory products, including antigen retrieval buffers for heat-induced or proteolytic-induced epitope retrieval. We also include hydrophobic pens in order to prevent any liquid running off the edges of your slides during staining, as well as H&E stains. So I want to provide a little bit more detail about our IHC kits. So these kits were designed to support the needs of our IHC users, ranging from beginners that have no prior IHC experience all the way to highly experienced users. So we offer three different types of kits. The first one is our IHC Easy Kit. These kits are ideal for those that are new to IHC or if you're trying to generate some quick data for a paper and you don't want to deal with, the troubleshooting aspect of it. So what these kits include are they're all going to be target specific, so they're ready to use, which means that they include a pre-optimized primary antibody along with all the reagents that are going to be necessary for you to complete your IHC workflow. We also provide an easy to follow protocol as well as an option to include a control slide for further validation. We also have our IHC prep and detect kits, and these kits are great if you're trying to develop an IHC workflow with a primary antibody that you already have or with one that you know that you're going to want to use. So these kits will include all the reagents that are needed for the IHC workflow except for that primary antibody. And then finally, we also have our IHC detect kits, which are going to be optimal if you already have an optimized staining workflow and you just need a kit for a highly sensitive chromogenic signal detection. We've also recently added H&E staining reagents to our offerings. So H&E is of course quite popular in the histopathology space. So in order to, visualize the structure and the organization of tissues. So the stains that we offer are going to be compatible with both paraffin fixed and frozen tissues, and they are conveniently ready to use. So there's not going to be any dilution or filtration that's going to be required. And these reagents are also available as individual stains, or you can also get them bundled as a kit. So now I want to switch gears a little bit. So I also want to highlight our multi-Rab goat recombinant secondary antibodies, which are validated for multiple applications, including IHC. So these secondaries are a multi-clonal mixture of recombinant monoclonal antibodies, which recognize multiple epitopes on the same IgG. So the idea behind this product was to generate a secondary that had the high sensitivity of polyclonals with the superior specificity of monoclonals. And because we are incorporating recombinant technology, our multi-Rabs have minimal cross-reactivity, unlimited supply, and high lot-to-lot consistency, which makes them ideal for reproducibility and long-term projects. So currently we have our multi-Rabs, which are conjugated to our in-house Coralite dyes, and this can be used for fluorescence IHC, or we also have our poly HRP multi-Rab, which allows for highly sensitive and biotin-free detection of primary antibodies in IHC. So unlike the avid and biotin-based signal amplification methods, the polymer HRP-based detection is going to offer advantages of lower background as well as a faster protocol, which is going to be particularly advantageous if you are using an autostainer for your IHC. And then finally, I just wanted to highlight our flexible antibody labeling kits, which allow you to label your own antibodies using a high affinity flex linker. So this is going to be particularly valuable if you need a conjugated antibody, but one is not commercially available for you. So what makes these kits so great is that they are compatible with any primary antibody. So the antibody does not need to be a protein tech antibody. It can be an antibody from any supplier. It also works with any antibody concentration and formulation. So, with traditional methods of antibody labeling, oftentimes you need to ensure that the antibody is in PBS only. And so, you'll need to do a buffer exchange. And so, during that process, you can lose quite a bit of antibody. So, it makes the entire process very expensive. But with these kits, you don't need to worry if your antibody has glycerol or azide or whatever in it. The kits will still work with it. It also allows you to label just as much as you need. So you can label as little as zero point 0.5 micrograms, or you can scale up if needed. So again, with traditional methods of antibody labeling, oftentimes you need to use the entire tube, and that can be a waste if you're only going to be doing your experiment a few times. It's also very fast and easy. So it's a 10-minute protocol. And the way that it works is you basically have your primary antibody, you add in the flex linker, and then within minutes, that flex linker will bind to the FC region of your primary antibody. You then add in a flex quencher, which binds to any of your unbound flex linker. And then that's it. Your antibody is labeled and ready to use for your assay. And so because you are pre-labeling these antibodies, they're really great for multiplexing using same species antibodies. So if you're trying to do fluorescence IHC and you have three antibodies that are all raised in mouse or all raised in rabbit, because you're pre-labeling, you can still use those antibodies on the same tissue. And as I mentioned, they are validated for IHC. They're also validated for IF, flow, as well as Western blot. And right now we have these different specificities, kits for these specificities and these different conjugates. So most of them are for our in-house correlite dyes, but we also have HRP and biotin as well. So with that, I just wanted to mention that you guys will have a couple follow-up emails coming your way. The first one will include a recording of this webinar, as well as a link to request one of our multi-rab recombinant secondary antibodies. So you can request a free sample. If you're eager to do that now, you can also scan the QR code here and do that right away. But you will also receive an email with the link as well. And then you'll also receive a certificate of attendance. And we do ask that you please allow 1 week for these follow-up emails to be sent. And so here I just wanted to put some contact information. So as I mentioned, there is a lot of questions in the chat and the Q&A box. So we probably will not get to all of them, but if you do have any further questions for us or for Laura, I've included our emails here, so you can always reach out to events@ptglab.com, or here I've also included Laura's email. Hopefully she's happy answering any questions that you guys might have. And if you liked today's webinar and you do want to learn about our upcoming workshops, please do follow us on social media because we post about all our upcoming events there. So with that, I will go ahead and start the Q&A. All right. So I jotted a few questions down that I think would be nice to start off with. The first question is asking you when you're using different types of tissues or their different sizes, do you ever change the temperature at all when you're sectioning?
No, I don't. Everything that's just me personally though, and the tissues that we get. So because we're quite a small lab, I don't have like it's also a human lab only. So any questions about I've seen some about mice and things like that. I do not know personally, just from my experience. But yeah, everything that I do is able to be done pretty much on the same temperature. I put my water bath up ever so slightly for biopsies, but no, everything I normally keep the same, and it seems to work okay.
Okay. And then a follow-up to kind of that, do you ever change the thickness of the samples? And if
So
so, what are
sorry.
the considerations? Sorry.
No, it's okay. So for immunos, we tend to cut everything on 4 microns, just because you want it thick enough to be able to see, but not massively thick. I think Congo red samples we would cut a little bit thicker, and as controls would reflect that. I tend to cut. Well, this is just seen as this is just about immunohistochemistry. I'll just keep it at immunos. Immunos I cut at 4. There might be some things with routine histology, so I tend to cut biopsies at 3. Skins, gallbladders, things like that are 3.5, and then breast lipomas, fatty tissue at 4. But yeah, immunos I keep all the same.
Great, thank you. We had another question asking if you had any tips when using the microtome and the tissues are falling off or they're breaking.
So I don't know if this, I think this might be one when someone said about the processing, which I would also say it could just be because your processing is quite harsh, or it's gone mushy. Maybe you've soaked it for too long, and it's gone like mushy and the tissue's just coming out. We find this in lets a lot. Your tissue might also just be really hard. So I would recommend that when you're trimming in or you're cutting, trim it in at a lower microns, and that'll just help a little bit. Yeah, I found it particularly with lets, it was quite difficult. So it could just be your tissue type. With regards to soaking, because I saw a couple of people asking about soaking, that is literally just like we would take our block once it's been embedded with the tissue in it and we trimmed it in to expose the tissue, we put it into water to soak it, and that just helps it stop being dry and brittle, and it means that you don't get the chatter that you see on your sections.
Great, thanks. The next question is asking, is there a good way to determine the proper clearance angle when the microtome hasn't been set up properly for a long time?
So I know it sounds like a cop out, but just get your engineers in to fix it. If you've got a service contract with an engineer, so we get ours serviced all the time. I say all the time, regular, and then we will just cut a practice block from this and make sure that everything's cut in the same. So yeah, that's just what you need to do to help with your clearance angle and making sure that your blade is in properly and it's been tightened enough. Yeah, finger tight, which I keep saying. That just means it just means you're not like proper forcing it or using any extra support. You're literally just going like as tight as your fingers can push it, but not overly tight. So yeah, that would also help just a bit.
Yeah, that's the one thing with like global talks and global webinars is a
Yeah,
lot of sayings don't
absolutely.
translate. Um, the next question is there a way for me to check for these artifacts before I do IHC? Just because IHC stains are costly to make a mistake.
They are absolutely. So some of them you'll be able to see on the water bath. So things like your carryover, or like any artifacts on the water bath, you might be able to see them. You might be able to see the folds as well. So it's completely up to you as the cutter to pick your most optimal section. So that is something that you're able to look at. But unfortunately, some of them like you're lifting things like that, you won't be able to see it until you've got the slides with you post immunos. But what you can do is just do a bit of trend analysis, like keep it like we have something where we write down, we've gone, this sample is lifted, this sample's lifted, or this sample's done that. And if we go, actually quite a lot of us ERs are lifting, that's when we then make the decision that rather than putting on the hot plate at 60 for half an hour, we're putting them in the oven for an hour. So just things like that would also be good. And then you'd be able to do just a little bit of like digging on why things are having issues. But yeah, unfortunately, some of them you won't be able to see until afterwards.
Great, thank you. The next question, I think we have time for maybe a couple more. I know it's at the hour, but we can do maybe two more. Can I use dab, stain, and H&E together?
Sorry, was that DAB and H&E?
Yes,
Yeah, I think we do. So we have our DAB kit and then we also have the hematoxylin to do the counter stain.
perfect. And then let's end with one more. So this person's saying that they are working with FFPE brain samples, mouse brain samples at postnatal days 0 to 10, which gives them a lot of autofluorescence after performing IHC with fluorescent antibodies. How can they avoid the autofluorescence?
This one I might not be great answering because I don't do fluorescence, so it's not something that I've actually had experience in, I'm afraid. I'm really sorry.
All right, no worries. So at Protein Tech, we would recommend trying to do some direct IF. So instead of doing like primary, secondary incubation, seeing if you can get a conjugated antibody that might help with autofluorescence. Other than that, you can try just, extending your blocking step to see if that helps get rid of any of the background. So let's do one more question then. There's a question saying that I sometimes catch bubbles of water under the wax when picking up sections. How can I prevent this?
So what I like to do is whenever I fill my water bath up and I see that the water bath is just getting bubbles in the bottom, we have like squeezy bottles for our water. So we have bigger bottles to carry more, but also squeezy bottles. And sometimes I literally just use the spouts of the squeezy bottle to like go along the bottom and get rid of all of them. Or depending on how deep your water bath is, you might be able to just get a slide and like scrape the bottom of the water bath to get rid of your bottles.
Great. Thank
Um,
you.
yeah, I think I've tried to answer a couple of questions in the chat, but I'm happy for anybody to contact me. I've put my Instagram handle in the chat, which I'm more able to respond to than emails. If you just let me know, like I watched the webinar and I just want to ask, especially if I've not asked the question, answered your question, please contact me and I will help absolutely anywhere that I can. So I'm really encourage you to contact me if that's okay.
Awesome. Thank you so much, Laura. We'll also include your email and your Instagram handle in the follow-up email as well. So if you haven't gotten that down, no worries about that. And you can always reach out to us as well, and we can always contact you with Laura too. So with that, I would just like to thank everyone for joining. Thank you again, Laura, for your wonderful presentation and for the great Q&A. And I will go ahead and end the webinar, and I hope everyone has a great rest of your morning, afternoon, or evening, depending on where you are tuning in from the world. So take care, everyone.